Hello and welcome back to the Biochemistry for Health Sciences channel. In the past few videos, we have been discussing some of the inorganic chemicals that have clinical significance. So in the previous videos, we talked about calcium. So today we'll talk about this inorganic chemical called phosphate. The phosphate or phosphorus test is one of the tests that is commonly ordered in the hospital, and you can see the value reference range is around 2.5 to 4.9 milligram per deciliter in case of this hospital. Always remember, this reference range is not the same from one hospital to the other because they use different assay kits, test kits. The way it's tested is different. Okay, so the population reference range might be different, and so on. So this reference range is just to give you an idea roughly what it is. But of course, you have to follow your own lab's reference range. In terms of millimolar, that comes out to be about 1 millimolar. So this serum phosphorus or serum phosphate test is one of the many chemistry tests that is often ordered in a clinical setting. So the phosphate, when it's not attached to any organic molecule, a carbon-based molecule, we call those phosphates inorganic phosphates, or sometimes represent them as PI, P for the phosphate, I for the inorganic. And you can see there, phosphate can have a variety of structures. These are all inorganic phosphates. And then there is another one called pyrophosphate that we represent PPI. There's basically two inorganic phosphates joined together. So the major species that you find in serum, where the pH is 7.4, is this one here, roughly about 80% of all the phosphate. And this monobasic form is less than about 20%. Now, why is that the case? So to understand that, let's start from here. This is phosphoric acid. We can see the H's are present, the hydrogens are present in all the three possible places. So as long as the pH is less than 2.2, the pKa, as long as the pH is less than this, less than 2.2, this would be the major form of phosphate that you would see in a solution. Once the pH is exactly 2.2, when the pH is exactly 2.2, same as the pK, then you have 50% of this and 50% of this. Okay. Once the pH goes greater than 2.2, now more and more of this will convert into this, the monobasic form. Okay, and then at pH 7.2, exactly 7.2, we have 50% of this and 50% of this. And once we go beyond 7.2, then we have more of this. Okay, and serum pH is 7.4. That's why this is the predominant form. And this is present in less amount. Now, once we hit a pH of 12.4, which you don't see in the human body, but in a solution, if you're doing an experiment, if you increase the pH to 12.4, now this will be 50% and this will be 50%. And once you go greater than 12.4, then the majority of the phosphate will be the tribasic form. Okay, so you can see, depending on the pH, you can have different types of phosphate. So for serum, this is the most abundant, and this is the second most abundant. Now I should tell you, because we will be using pKs a lot in the future videos, especially with proteins, it's very, very important to understand this process by which the acids lose their protons, okay, lose their hydrogens. So this is a very important concept. pK values are given to you. You can see that in books and all that, but be warned that pK values can change. Okay, they can change depending on the environment. So these pK values are just to give you an idea of what you can expect to see. So not only does your body have inorganic phosphates, our cells also have a lot of what we call organic phosphates. Organic phosphates, very, very, very common. So basically what we have, this is the type of reaction we see a lot. So let's say there's a small organic molecule, some carbon-based molecule, and it has a group called hydroxyl. This is an organic chemistry group called hydroxyl. And then in the presence of something that can donate phosphate, commonly ATPs, the energy molecule is also used to do these reactions that we call phosphorylation or phosphoryl transfer. So this phosphate, one of these phosphates can be transferred to this OH. And then we get what is called, you see, a COPO. This is called a phosphoester. Phosphoester. Okay, organic phosphates in the form of phosphoesters are very, very common in the cells. Examples, of course, a lot of energy molecules, ATP and creatine phosphate, many metabolic intermediates that will come across in metabolism, such as glucose 6-phosphate, pyranol phosphate, you can see all the P here, phosphate dihydroxyacetone phosphate, isopentenyl pyrophosphate, and many examples. Then there are several coenzymes. These are molecules needed by certain enzymes, very important in metabolism. NAD, FAD, NADP, coenzyme A, thiamine pyrophosphate, PLP, and so on. Many coenzymes require or have, or they are basically organic phosphates. So very common in our cells. So we not only have small organic phosphates, many of our proteins, is estimated that about 30% of the proteins in our cells at any one time are phosphorylated. So why does this happen? Why do proteins get phosphorylated? Remember I told you proteins work by changing shapes. Their functions happen because they can change shapes. So shape, three-dimensional shape is very important for a protein. So here's a protein, and let's say it gets phosphorylated, okay, with the help of some phosphate donors such as ATP. And these are all catalyzed by enzymes we'll talk about later. These enzymes are called kinases. The phosphate is transferred to this OH group, and you get a phosphate ester. Now notice this phosphate has two negative charges. So negative is going to repel some negative charge on the protein. So here's a negative charge on the protein, but the negative is going to attract some positive charge on the protein. Okay, so because of this electrostatic repelling and electrostatic attraction, the protein shape changes. And once the protein shape changes, it changes the activity of the protein. The protein may become a better, if it's an enzyme, it may become a better enzyme. If it's a transfer protein, it may become a better transfer protein, or it might become a worse enzyme or worse transfer protein. So the shape change can either increase the activity or decrease the activity. Protein stability, other properties can change. So stability of a protein might change when it's phosphorylated. It might become more stable or less stable. the movement or the transport of the protein might change. Okay, phosphorylated proteins may not enter a certain compartment, such as a nucleus. And we see that a lot in transcription factors, and we'll look at that later. The interaction of the proteins with other proteins will change, with lipids, protein interaction with lipids, protein interactions with DNA, RNA, and other molecules are going to change because the shape has changed. And then once the protein is negatively charged, because phosphate groups are negatively charged, okay, remember we're not just adding one phosphate group. A protein may have 2 phosphate groups, or 3 phosphate groups, or 25 phosphate groups, or 67 phosphate groups, or 79 phosphate groups. We can have many phosphates added to the protein so that the protein changes shape little by little by little. And that's the remarkable thing about our proteins, is not only can they do a function, their function can be very finely tuned, very finely regulated. Okay, so all the negative charges on the protein is going to make the protein, so let me draw the protein here, basically negatively charged, and membranes themselves are negatively charged. So when a protein tries to get out of the membrane by exocytosis or whatever, if the cell doesn't want it to get out, it's not going to go through. Okay, because negative repels. Okay, so it's going to keep all the proteins inside the cell. So where is your phosphate located? Not surprising, we talked about calcium phosphate before. Most of your phosphate is in the skeletal system, over 85%. The rest are inside cells, about 14%, and then extracellular outside cells is very little, about 1%. So serum, we have the inorganic phosphates, and we might have a little bit of phosphates in the lipoproteins. So in the cells, you can see your phosphates. You can see all the small molecule phosphates, nucleotides, and other organic phosphates. Large molecule phosphates, such as your phosphorylated proteins. Membranes, there are many phospholipids. We'll talk about this later. Phospholipids are an important component of membranes. And you can see in these phospholipids the presence of phosphates. And then, of course, we'll see in a few seconds, nucleotides, which make up DNA and RNA. The RNA and DNA are all phosphoesters. Okay, so phosphates are used, they're very stable compounds. They have special chemistry properties that make them very good for energy transfer. And it's all about energy. Remember, we talked about this in an earlier video. Energy. And they're also very good for information. Phosphate esters are very good for information. So we have enzymes that can use these phosphate esters for energy transfer and can use this use these phosphate esters for information transfer. So probably one of the best information transfer that we all heard of is our DNA. Okay, so here's a cartoon of a DNA molecule, and you can see the phosphate, the sugar, the phosphate. You can see a lot of phosphates. Okay, these phosphoesters are very, very stable, and we have so many enzymes that can manipulate these DNA, RNA, and other information molecules, so that we can use this DNA to make our RNA, and then eventually to make proteins, and then the protein will carry out the cellular activity, and all specialized, 200 specialized cells in your body will give you your anatomy and your physiology. Okay, so very stable molecules, phosphoesters. So that's why nature decided to use phosphate. Very complex chemistry. We still don't know much about a lot about the phosphate chemistry, but very, very fascinating chemistry that is related to biology. Okay, so we will stop here, and thank you for watching, and we will see you in the next video.